Hello dear friends and a warm welcome. This is the part 3 of multiple choice questions on microbiology and applied microbiology. This is the question paper of the MSc entrance exam conducted by Banaras Hindu University. Now this exam is conducted by the NTA under the CUET PG exam. This part 3 contains question number 101, question 150. The first 100 questions have been already discussed in the part 2 and the part 1. The link for the part 1 and part 2 you will get in the description box below and in the I button here. This video is very important for you if you are preparing for the competitive exam. So watch this video till end. Myself Dr. Tripti Malika Ahuja. I am your tutor, guide and mentor for the subject biology, biological sciences, life sciences and microbiology. In the playlist, you will find there a number of videos which will help you in the preparation of your exams. So if you are new to this channel, then do subscribe it and don't forget to press the bell icon so that you can get the notifications of all the new videos. Before I start, I suggest you to take out a paper and a pen. I will read the question and the 4 option. Out of this, one option which is correct, which you think is correct, you can write it. Then I will tell you the correct answer with an explanation. So that will help you in solving such questions. So question number 101. What is pyrogen? Options are a class of bacteria, a disease, toxins produced by pathogen, none of above. Pyro means heat. Gen means generating substance. So pyrogens, these are the substances which are produced by some bacteria. These are the toxins produced by particularly the gram-negative bacteria. So the option 3 is correct here. 102. What is opportunistic infection? 1 infection in ill person, 2 infection in healthy person, 3 infection caused by other than pathogen, 4 disease. The option 1 infection in ill person is correct. Opportunistic infections, these are the infections which are caused by the opportunistic pathogens. The pathogens which are usually non-pathogens and they are called opportunistic pathogens. They seldom cause the infection in the normal individual. Next question is the question number 103. Reason for immunodeficiency is 1 recurrent infection, 2 malnutrition, 3 cancer chemotherapy, 4 all of the above. The option 4 all of the above is correct. Immunodeficiency means the immunodeficient person, the immune system is not working properly. So all of these conditions, they are responsible for the malfunctioning or the deficiency of the immunity. Question 104. Which is a waterborne disease? 1 amoebiasis, 2 trichomoniasis, 3 leishmaniasis, 4 none of the above. The option 1 amoebiasis is correct. Amoebiasis is caused by the protozoan and amoeba histolytica, which is acquired by drinking the water which is contaminated. Question number 105. African sleeping sickness is caused by 1. Entamoeba histolytica, 2. Giardia lamblia, 3. Trypanosoma brucei, 4. Toxoplasma gondii. Out of this, the option 3, Trypanosoma brucei is. Question number 106. Which is not a protozoal disease? 1 malaria, 2 amoebiasis, 3 gingivitis, 4 none of the above. The option 3, gingivitis, is correct. Gingivitis is caused by bacteria like Streptococcus, Fusobacterium actinomyces. Question number 107. Method for viral diagnosis is 1 gel electrophoresis, 2 polymerase chain reaction, 3 sequencing of genome, 4 all of the above. The correct option is the 4 all of the above. Question number 108. Which is not belong to the same class? 1 bacteriophage, 2 staphylococcus, 3 clostridium, 4 none of the above. Now this question is somewhat a different question and not very clear question. So here 1 bacteriophage virus, 2 and 3. Staphylococcus and clostridium, these are bacteria. So viruses, they are different. So we can say 1 bacteriophage is the correct answer. Question number 109. A protozoal disease caused by sexual contact. 1. Trichomoniasis, 2. Leishmaniasis, 3. babiosis, 4. None of the above. The option 1, trichomoniasis, is correct. It is acquired by Trichomonas vaginalis. Question number 110. Which is not related term with STD? 1. Sexually transmissible disease, 2. Sexually transmitted infection, 3. Venereal disease, 4. All of above. Now this question is incorrect because all of these three, sexually transmissible disease, sexually transmitted infection and the venereal disease, these are same. They are the diseases which are transmitted sexually. So I could say that the question should be which is related term with STD, then the option 4 would have been correct. Next question is the question number 111, which is not a antimicrobial agent. 1. Antibiotic, 2. Analgesic, 3. Antifungal, 4. Antiparasitic. Antibiotics are against the bacteria, antifungal against the fungi, antiparasites against the protozoans and the parasites, but analgesics, they are used to cure the body pain. So the option 2 is correct. 112. Which class of antimicrobial is not used in living object? 1. Antibiotics, 2. Disinfectant, 3. Antiviral, 4. All of the above. The option 2, disinfectant, is correct. For example, phenols. 113. What is the reason of drug resistance? 1. Excessive use of drug. 2. Prescribing low doses. 3. Self-medication. 4. All of the above. Drug resistance is the case when the drug which was initially active against the pathogens now does not act against those pathogens. So here the option 4 is the correct answer. What is the meaning of term refractory? 1. Reflex action, 2. Reference drug, 3. Patient failed to respond to a drug, 4. Response of a drug. Here the option 3, patient failed to respond to a drug, is the correct. The term rhizosphere was coined by 1. Klopler, 2. Weller, 3. Lister, 4. Hiltner. Here the correct one is the 4. Hiltner. The full name is the Lawrence Hiltner. The rhizospheric bacteria or the rhizosphere means the soil which are adjacent to the plant root. Rhizospheric bacteria which are present in the soil adjacent to the roots of the plant. 116. Which of the following microorganism can be used as a biofertilizer? 1. Pseudomonas, 2. Salmonella, 3. Agrobacterium, and 4. Lactococcus. 1. Pseudomonas is the correct. The Pseudomonas, these are used to increase the soil permeability, fertility of the soil by the demineralization process. Question 117. PGPR is 1. Plant growth preventing rhizobacteria, 2. Plant growth hormone producing rhizobacteria, 3. Plant growth promoting rhizobacteria, 4. Popular growth promoting rhizobacteria. The correct one is the 3. Plant growth promoting rhizobacteria. Question number 118. Bacteriocins are produced from species of 1. Pseudomonas, 2. Lactobacillus, 3. Agrobacterium, 4. Clostridium. Here the correct answer is the Pseudomonas. Bacteriocins, these are the substances which are produced by a bacteria which inhibit the other closely related bacteria. The bacteriocins which are produced by the Pseudomonas, these are called the pyocins. 119. The nitrogenase enzyme complex is comprised of 1. Dinitrogenase, 2. Dinitrogenase reductase, 3. Both 1 and 2. 4. Nitrogenase carboxylase. The correct one is the both 1 and 2. The option 3. Nitrogenase enzyme is responsible for the conversion of the ammonia to the nitrogen. The VAM is actomycorrhiza, 2. Basidiomycetes, 3. Actinomycetes, 4. Endomycorrhiza. The correct one is the 4th option, which is the endomycorrhiza. These are the fungi which are found in the roots of the higher plants. Question number 121. Pesticides include 1. Fungicides, herbicides, insecticides, nematicides, rodenticides. 2. Insecticides only. 3. Herbicides, insecticides and nematicides. 4. Insecticides, nematicides and rodenticides. Here the option 1, fungicides, herbicides, insecticides, nematicides and rodenticides is correct. Question number 122. The common mode of action of most insecticides is through the attack on 1. Respiratory system, 2. nervous system, 3. muscular system, 4. circulatory system. The option 2, nervous system is correct. Most of the insecticides, they act on the nervous system, causing the potassium and sodium imbalance in the nervous system. Next one, 123. A carbamide pesticide is 1. propoxur, 2. simazine, 3. atrazine, 4. tetraethyl pyrophosphate. Here the correct one is the first one, which is the propoxur. Propoxur, these are the esters of the N-methyl and N-dimethyl carbamic acid. Other examples are LT-carb and the pterygocarb. Question number 124. The chemical fertilizer required for better rhizobial nitrogen fixation is 1. phosphorus, 2. potassium, 3. calcium and 4 is sodium. The correct one is the first, which is the phosphorus. Question number 125. A green manure is 1. rice, 2. sorghum, 3. maize, 4. sesbania. Here the correct one is the sesbania. These are the plants which can grow in the fields and they enrich the soil with the nitrogen and the sulfur. For other examples of the green manures are the cannabis, clover, beans, peas, ryegrass and some grasses. 1 to 6. Azotobacter and Bacillus polymixa are 1. decomposer, 2. non-symbiotic nitrogen fixer, 3. symbiotic nitrogen fixers, 4. pathogenic bacteria. Here the correct one is the 3 symbiotic nitrogen fixers. They fix the nitrogen in the rhizospheric soil. Question number 1 to 7. Triticale is man-made cereal obtained through hybridization between wheat and rice, 2. wheat and rye, 3. wheat and oat, 4. wheat and gram. The option 2, wheat and rye is correct. Question number 128. Producing new plants by cells instead of seeds is 1. tissue culture, 2. biofertilizer, 3. amphimixes, 4. miro mixes. The correct one is the first, which is the tissue culture. The phenomenon of using a predator for controlling a pest is 1. biological control, 2. genetic engineering, 3. artificial control, 4. confusion technique. This one is the first, which is the biological control. For example, gambusia fish, it is used to control the larvae of some mosquitoes. Question number 130. Source of Bt gene used for production of transgenic plants is 1. Pseudomonas, 2. Brachybacillus, 3. Lactobacillus, 4. Bacillus. The correct one is the 4. Bacillus. It is actually Bacillus thuringiensis. Question 131. Genes for biodegradation of xenobiotics by Pseudomonas are located on 1. plasmids, 2. genome, 3. transcriptome, 4 is the proteome. The option 1, plasmids is correct. Plasmids are the extra chromosomal segments which are found in certain bacteria. Question 132. The first stable product of nitrogen fixation is 1. nitrite, 2. nitrate, 3. ammonia, 4. nitric oxide. 3. ammonia is the correct answer. Next question 133. Bread mold is Saccharomyces cerevisiae, 2. Rhizopus stolonifer, 3. mucorrhizae, 4. Streptococcus pneumoniae. The option 2, Rhizopus stolonifer is the correct. The black bread mold, it belongs to the class Zygomycota. Question 134. Which of the following is not used as food preservative? 1. citric acid, 2. lactic acid, 3. propionic acid and 4 is the picric acid. The option 4, picric acid is correct. The picric acid, these are harmful for the skin and also toxic upon inhalation, so they are not used for food preservation. 135. Microbial quality of milk is generally estimated by 1 MBRT, 2 culture techniques, 3 turbidity test, 4 ambient test. The first one, MBRT, is the correct one. MBRT stands for the methylene blue reduction test. 136. Ultra high temperature UHT milk is pasteurized at 133 for 2 seconds, 140 to 150 degrees Celsius for 1 to 5 seconds, 300 degrees Celsius for 5 minutes, 400 degrees Celsius for 10 minutes. The correct one is the option 2. The option 2 is the 140 to 150 for 1 to 5 seconds. Question number 137. Staphylococcal gastroenteritis is 1 food poisoning, 2 cancer, 3 infection, and 4 is the food disorder. The correct one is the first, which is the food poisoning. Question number 138. C1, C2, C3 are produced by Staphylococcus aureus. They are 1 alpha toxin, 2 endotoxin, 3 enterotoxins, 4 are the viral toxins. The correct one is the 3 enterotoxins. Question 139. HACCP system was developed by 1 MSc MCF, 2 FDA, 3 WHO, 4 UNESCO. The HACCP system was originally developed by the NASA, National Aeronautics Space Administration, under the FDA, Food and the Drug Administration of USA, and it is for the control of the biological, chemical and the physical hazards concerned with the food items. Question 140. So here the correct answer is the 2 FDA. The question 140, pH of milk is 6.1, 6.2, 6.6, 6.3. The correct one is the 3, 6.6. Question 141, the decomposition of animal proteins by anaerobic microorganism is known as 1 ropiness, 2 moldiness, 3 putrefaction, 4 sliminess. The correct one is the 3 putrefaction. Question number 142, toxin produced by Clostridium botulinum is 1 enterotoxin, 2 exotoxin, 3 neurotoxin, and 4 is mycotoxin. The correct one is the 3, which is the neurotoxin. Question 143. The organism responsible for yogurt formation is 1 Streptococcus salivarius, 2 Streptococcus salivarius, species Thermophilus, 3 Lactobacillus thermophilus, 4 Streptococcus bulgaricus. The correct one is the Streptococcus salivarius, species Thermophilus. Question 144. The most common qualitative test carried out for foods is 1 aerobic plate count, 2 measurement of ATP, 3 using direct filter technique, 4 direct microscopic count. The option 1 aerobic plate count is correct. 145. Botulism is caused by 1 Staphylococcus aureus, 2 Clostridium perfringens, 3 Clostridium botulinum, 4 Salmonella typhi. The correct one is the 3 Clostridium botulinum. Question 146. Tempeh is produced from 1 Rhizopus oligosporus, 2 Lactobacillus plantarum, 3 Pediococcus halophilus, 4 Pediococcus acidilecti. The option 1 is correct, Rhizopus oligosporus. This tempeh is a soybean traditionally fermented using the Rhizopus fungi. 147. Milk spoilage is caused by species of 1 mucor, 2 bacillus, 3 leuconostoc, 4 yeast. Here the correct one is the 3 leuconostoc. In addition to leuconostoc, enterococcus, lactobacillus, lactococcus, they are also responsible for the spoilage of milk. Question 148. Koji process is used for the production of 1 soy sauce, 2 tempeh, 3 cheese, and 4 is the sauerkraut. Here the correct one is the 1 soy sauce. Koji process is the process which is applied to the proteins. Here there is the koji acts like a curing agent also in the fermentation of the coffee beans, and also for the production of the soy sauce. Question 149. Milk is pasteurized at 1. 70 degree for 20 minutes. 2. 65 degrees Celsius for 10 seconds. 3. 72 degrees Celsius for 15 seconds. 4. 80 seconds for 20 minutes. Here we can say that the option 3, 72 degrees Celsius for 15 minutes is correct. The original method was the pasteurization at the 62.5 degrees Celsius for the 30 minutes. Now there are also the high temperature and the shorter holding times, for which the 72 degrees Celsius for 15 seconds is the most appropriate here. Last question, the question number 150 is, in HACCP, P stands for 1. Hazard analysis critical control pasteurization. 2. Hazard analysis critical control points. 3. Hazard analysis critical control pressure. 4. Hazard analysis critical control product. So here the option 2, hazard analysis critical control points is correct. This is a systematic approach for identification, evaluation and control of the food safety hazards. So these were the 50 questions which will help you in the preparation of your exams. Best of luck for your exams. Bye and take care.